Okay, before we get going, are there any questions? No questions. All right. Let me mention a little bit about some assignments. Remember in the labs that the labs will be graded twice. The first time you will either get the first time it's graded, you'll either get the full credit for the lab, which for the labs now, that'll be 5 points, or you'll get 0.01. That 0.01 is just a code to tell me to go back and check that students may submit corrections. So that is not your grade. That's a code to tell me to go back and check that assignment. Let me mention that, oh, where do I have that? Come on. My computer's a little slow for some reason. Let me remind you that Lab 5 and Lab 6 have been graded the first time, and you have until this Saturday to submit corrections. And you can submit corrections to something like 11:59 p.m. Saturday night. Any questions about any of that? Why do I have so many Zoom meetings open? Let me go ahead and close that. All right. I think that's all I wanted to say about the lab and the gradings. I do have graded the last lecture worksheet, which I don't remember which one that was. Let me look here. Come on. My mouse isn't working. Maybe lecture 4B. Yes. And then I think it's lecture 8. Is that the next lecture we're working on? That one is due this Saturday. Let me shut the grade book and then I'll show my screen. Now, why did that pop up? It's a little slower because my mouse isn't working. Come on. I think that's the assignment we're on. We might be on chapter 5. I don't remember. Let me go down here. Yeah, we're in chapter 5. I opened the wrong PowerPoint. So the chapter 5 lecture worksheet will be due this Saturday. All right, any questions before we get started? So we're going to be covering chapter 5 today, and hopefully we'll finish it and start chapter 6. In the lab today, we're going to be covering lab 7. And you'll, well, no, you're not really, it's doing unknown work. Your unknown work, you have 3 projects which are started, and actually only the gelatin hydrolysis has been started. Come to the assignment. So we had talked about the different factors that can influence enzyme activity. And the last factor we talked about was feedback inhibition. I need to talk a little bit about enzymes. Most enzymes are proteins. All of the enzymes that you probably are familiar with are proteins. However, some enzymes are RNA molecules, and these are called ribozymes, where they are RNA enzymes. The only one that we'll talk about in this class is actually when the ribosome is removing the amino acid from tRNA and connecting it to the growing chain of amino acids to the growing chain of the protein. That enzymatic activity is done by an RNA enzyme. And that's the only one we will talk about in this class. But there are other RNA enzymes, which the book calls ribozymes, that cut and splice RNA molecules or other molecules. They function as enzymes in that they have active sites that actually bind to the substrates. And they're not used up or modified in a chemical reaction. The only difference is instead of being made up of protein, they're made up of RNA. And like I said, that one example, we're going to talk about the ribosomes. That is not mentioned in your book, but I do mention it briefly in the lecture. So when we're talking about organisms, we find that they have a lot of diversity, and they can have metabolic diversity as well. All organisms, including microbes, require an energy source and a carbon source. We can divide organisms, we're talking about microbes in this case, by where they get their energy source and where they get their carbon source. So if we divide the organism on where they get their energy source, the organism can be classified as a phototroph or as a chemotroph. Phototrophs derive their energy from light. So these are the photosynthetic organisms. Chemotrophs derive their energy from either an inorganic or an organic chemical compound. And we are a chemotroph. We derive our energy from an organic compound. Someplace I have an apple here, I'll show you there. There we go. My energy source. Any questions? You guys are supposed to remind me if I don't have my headphone on to put on the headphone. Let me state that there's a big difference between whether you derive your energy source from an organic chemical compound, hopefully you can see the apple here, or from a molecule like glucose, which is also an organic chemical compound, as opposed to an inorganic compound, such as sulfur, or a nitrogen compound, or iron. The organisms which can get their energy needs from an inorganic chemical compound are mostly prokaryotes, and what they are the chemosynthetic bacteria, deriving their energy needs from a chemical compound. Any question about any of that? You can also classify organisms on where they get their carbon source. Organisms can be classified as autotrophs or as heterotrophs. Autotrophs get their carbon source from CO2, dissolved in either the air or the water. And what is an autotroph? Give me an example. Come on, I'm not hearing anyone.
Maybe like algae?
Can you repeat that? I didn't hear you.
Algae?
Oh yeah, green algae or even brown algae or red algae. All of those are photosynthetic organisms. And what's the most widely known photosynthetic organism? Come on, it grows on the land.
Plants.
Plants, thank you. Heterotrophs, on the other hand, use an organic chemical compound for their energy needs. So humans are a heterotroph, and we derive our carbon from drink, largely glucose, but it can be other molecules as well. All right, can somebody confirm that you can hear me? I just plugged in my headphone.
I
Yep.
can hear you.
Okay, great. Now, we can combine where an organism gets its energy and carbon source and then classify the organism on the combination where they get both their energy source and their carbon source. And if this gets a little confusing to you, remember to just take it a step back and say, okay, what's the energy source? If it gets it from light, it's a phototroph. If it gets its energy source from a chemical, it's a chemotroph. And then say, what is its carbon source? And if it gets its carbon from CO2, it's an autotroph. That means they literally can make their own food. And if they get their energy needs from an organic chemical compound, they're a heterotroph. Altogether, we have photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs. I'm going to discuss photoautotrophs first because that's the easiest, well, one of the two easiest to discuss. Photoautotrophs get their energy needs from light, why they're called a phototroph, and then get their carbon source from CO2, why they're called an autotroph. So photoautotrophs. And green plants and algae and green photosynthetic bacteria are photoautotrophs. Any question about that? Now, this slide is telling you that we could separate the photoautotrophs into two types, the oxygenic and the anoxygenic, but I'm not really going to test you on oxygenic phototrophs. That means that's something that gives off oxygen, and an anoxygenic phototroph, that's something that does not give off oxygen. It gives off another what do you call it? Another molecule, and it's usually sulfur dioxide. You don't need to know that, but I guess the only thing you should know is that there are two types of photoautotrophs. Now I'm going to skip a photoheterotroph and a chemoautotroph. We'll come back to them. The next easiest to discuss is a chemoheterotroph, and humans are chemoheterotrophs. We get our chemical needs for energy from a chemical compound, and that's why we're a chemotroph, okay? And then we get our carbon source from an organic chemical compound, and so we are a chemotroph. And put them together, it's a chemoheterotroph. We get our energy needs from a chemical compound and our carbon needs from an organic chemical compound, okay? And oftentimes for chemoheterotrophs, especially for humans, we get both the carbon source and the energy source from the same molecule. So like, for example, if you're eating glucose, you're going to get both your carbon needs and your energy needs from the glucose. Any question about that? All right. Now, there are some chemoheterotrophs which get their, let's see, is it energy needs? Gets their energy needs from a inorganic chemical compound. Usually they get their carbon needs from an organic chemical compound, but for their energy needs, they can get it from an inorganic chemical compound. These are some of the photosynthetic organisms. Not photosynthetic, excuse me, the chemosynthetic organism. Some of the chemosynthetic organisms are chemoheterotrophs. If they get their energy needs from an inorganic chemical compound, but they get their carbon from an organic chemical compound, they are a chemoheterotroph. And like I said, some of the chemosynthetic bacteria are chemoheterotrophs. Any question about any of that? All right, let's move on to the chemoautotrophs. They get their energy needs from a chemical compound, and they get their carbon dioxide needs from CO2. So they're a chemoautotroph. Some of the chemosynthetic bacteria are chemoautotrophs. Remember, some of them, some of the chemosynthetic bacteria are chemoheterotrophs. The difference is what their carbon source is. If they can get their carbon from CO2, they are a chemoautotrophs. So the chemosynthetic bacteria are split into two groups, the chemoheterotrophs and the chemoautotrophs. And the devising agent is where they're getting their carbon needs. If they get it from CO2, they're a chemoautotroph. If they get it from an organic chemical compound, they're a chemoheterotroph. You actually are familiar with the chemoautotroph, you just don't know it. If you ever lift up the toilet tank, I'm not talking about the toilet bowl, I'm talking about the container that holds the water that you flush into the toilet bowl. The toilet tank, you know, all that brownish or yellowish scum attaching to the back of the toilet tank? That is chemoautotrophs, largely chemosynthetic bacteria that are getting their energy needs from a chemical compound dissolved in the water. And it could be iron, it could be sulfur if you have sulfur in your water. Most people don't, in which case it's iron. And then it gets its carbon from CO2 in the air or dissolved in the water. So you all know a chemoautotroph. You just didn't know it. Any questions about any of that? All right, now let's talk about the photoheterotrophs. The photoheterotrophs get their energy need from light. That's why they're called the phototroph. And they get their carbon source from an organic chemical compound. There's probably no student who knows a photoheterotroph. If you do, tell me I'm wrong. The green and purple non-sulfur bacteria are photoheterotrophs, getting their energy needs from light. So they are photosynthetic, but they do not get their carbon from CO2. They get it from an organic chemical compound such as glucose. And most of you who probably never heard of the green and purple non-sulfur bacteria. Okay. All right. Any questions about these groups? Photoautotrophs get their energy needs from light, carbon source from CO2, and green plants are photoautotrophs, as well as green photosynthetic bacteria like the cyanobacteria. The photoheterotrophs, nobody knows these bacteria, but they get their energy needs from light And then they get their carbon source from an organic chemical compound such as glucose. We do not even grow these in a microbiology lab. They're actually difficult to grow, although you can use a specific media and then grow them. Chemoautotrophs, like I said, you actually are familiar with one, and they get their energy needs from a chemical compound. The one in the toilet tank gets it from iron, but it could be any organic chemical compound like hydrogen sulfide, sulfate, nitrogen compounds. And they get their carbon source from CO2. The chemoheterotroph should be familiar to you because we're a chemoheterotroph. We get our energy needs from a chemical compound such as glucose, and our carbon source from an organic compound such as glucose. And oftentimes chemoheterotrophs get both their energy source and their carbon source from the same compound, but it doesn't have to be. Like I said, some of the chemosynthetic bacteria get their energy needs from an inorganic chemical compound, but they get their carbon source from an organic chemical compound. And that would be a chemosynthetic bacteria that's not a chemoautotroph. All right, any question about any of that? I guess I can say that all animals are chemoheterotrophs, the protozoa, all the fermentative bacteria. Many of the bacteria we grow in the lab are chemoheterotrophs, like E. coli, fungi like mushrooms, yeast, mold, or chemoheterotrophs. All right, so here's the nutritional classification of all organisms. You should know everything in red. We can divide all organisms by their energy source. If they get their energy from a chemical compound, we call them a chemotroph. If they get their energy source from light, they're a phototroph. We can then divide those by where they get their carbon source. If they're a phototroph and they get their carbon from CO2, they're a photoautotroph. If they get their carbon from an organic chemical compound such as glucose, they're a photoheterotroph. Okay, and I did mention that the photoautotrophs can be split into the oxygenic and the non-oxygenic, or the anoxygenic. You can divide the chemotrophs by where they get their carbon source. If they get their carbon source from an organic chemical compound such as glucose, they're a chemoheterotroph. And if they get their carbon source from CO2, they're a chemoautotroph. You should know that the chemoheterotrophs are of two very different groups. And that is, it's not even shown here, that if they, where am I losing it here? Oh, here it is here. And that is that you could break it up here. If it's an organic chemical compound, it's a chemoheterotroph that you know. And if it comes over here and it's an inorganic chemical compound, it's chemosynthetic bacteria. Okay. Any question about that? All right. Now, this neat classification system, I have to tell you, is not always so clear cut. There are complications. An organism can actually change its classification depending on what is available. Euglena, when light is available and no carbon source is available, will actually act as a photoheterotroph, making its food source from light and using CO2. Excuse me, it'll act as a photoautotroph. Okay, using light energy and CO2 for its energy needs and its carbon source. However, if you put glucose in the media, euglena can then act as a photoheterotroph, using light for its energy needs, but then using glucose for its carbon needs. Okay? And we're not going to talk about why the organism does that. It's just more efficient using glucose than using CO2. But if we were to take out the light and have glucose in the media, euglena will then act as a chemoheterotroph, using the glucose for its energy needs and using the glucose for its carbon source. Euglena does not act as a chemoautotroph, and that is, if glucose is available, it'll use it for its carbon source. It won't use CO2. But we can talk about some of the chemosynthetic bacteria, and they will use whatever is available. So the chemosynthetic bacteria are chemotrophs. They get their energy needs from a chemical compound. But if CO2 is only available, there'll be a chemoautotroph. However, if glucose is available, they can act as a chemoheterotroph. At the same time, they're acting as a chemoautotroph, meaning they will use both CO2 and glucose for their carbon needs. And we call these organisms mixotrophs. They will use whatever is available. There are some photosynthetic bacteria which can act as a photoautotroph and as a photoheterotroph, and they may do it at the same time. Okay, and that is CO2 is available, so there will be a photoautotroph, but if glucose is available, they can be a photoheterotroph. And these are, like I said, just mixotrophs. They use whatever is available. I know it best for the chemosynthetic bacteria. Any question about any of that? All right. We talked about energy, where organisms get their energy source. Let me say that nutrient molecules have energy in them, and an organism uses that energy by breaking the bonds of that molecule. So the nutrient molecule has the energy associated with the electrons forming the bonds of that nutrient molecule. And so when we get our energy need from glucose, what we do is we break the bonds of glucose. In cells, energy is often obtained from catabolic reactions. And then it's stored, that energy is stored in the bonds of ATP. For all of the chemoheterotrophs, they get their energy needs from catabolic reactions. And much later on, we'll actually talk about the photosynthetic bacteria. They can get their energy needs from catabolic reactions. Whenever the light is not available and the plant wants energy, it will use a catabolic reaction to get the energy. Any questions about any of that? I briefly mentioned energy is stored in the bonds of ATP. ATP is the energy carrier of cells. Generally speaking, whenever a cell wants energy for its needs, it will get that energy needs from ATP. ATP has high energy in unstable bonds. The third and the second phosphate group is a high energy bond. And the molecule can be broken apart to release that phosphate. And then that will release a large amount of energy, which can be quickly and easily used by the cell for its energy needs. Any question about any of that? How are you guys doing?
Good.
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 All right. Let's talk about how the cell derives energy. And this is not true for photosynthetic organisms, but it is true for most organisms. And the start of this energy production does occur in even photosynthetic organisms. But in chemoheterotrophs, they mainly get their energy needs from catabolic reactions, breaking down a larger molecule into smaller molecules. And they produce energy by oxidation reduction reactions. Oxidation is the loss of electrons. So molecule A loses an electron, and so we say it is oxidized. And then the electrons move to molecule B. Molecule B, we say, is reduced. Reduction is the gain of electrons. Oxidation reduction reactions are always coupled, meaning one molecule or atom loses the electron, the other molecule or atom gains the electron. Now, oxidation reduction reactions is a mouthful, so they came up with a shortening, and they call it a redox reaction. It means the same thing as oxidation reduction. An example is copper plus iron 3 pluses, meaning it has 3 positive charges, can react to become copper 2 plus. Copper lost an electron, so it becomes more positive, and we say it was oxidized. And then the iron gained an electron, so it's reduced, and it loses its positive charge down to 2 plus. Any question about any of that? An easy way to remember oxidation reduction reactions is you can say, Leo the lion goes grr. Leo, lose electrons, oxidation. Grr, gain electrons, reduction. So remember, Leo the lion goes grr. Any questions? Now, in biological systems, oxidation reactions usually have the proton, that's an H plus ion, that is removed with the electrons. Remember, in the hydrogen atom, it is an H plus ion plus an electron with a negative charge equals the hydrogen atom. And that's all the hydrogen atom is, at least the simplest form of hydrogen. So in biological systems or biological oxidations, we often call these reactions dehydrogenations because besides the electron being lost by a molecule and gained by a molecule, the hydrogen is also lost by this molecule, why it's called dehydrogenations. And then the other molecule gains the hydrogen as well as the electron. Okay? Any question about that? So actually in our cells, this organic molecule would be glucose, and it loses both a hydrogen ion and an electron, which is gained by NAD+, which is a coenzyme and an electron carrier. We'll talk a little bit more about that. And that becomes an oxidized molecule. So let's see, that would be glucose minus 1 hydrogen. So it would have a negative charge. And then NAD+ gains a hydrogen ion and 2 electrons. And so it becomes NADH. Any question about that? Okay, biological redox reactions are used in catabolism to extract energy needs from nutrient molecules. So we use redox reactions to break down like glucose, so we can then use the energy in glucose. Highly reduced compounds, that is, compounds with many hydrogen atoms, are degraded, that means catabolized, and changed into highly oxidized compounds. For example, glucose, C6H12O6, has a lot of hydrogen, so it's a reduced compound. It's oxidized into the molecules of CO2 and water. And in reality, I should probably put 6 carbon dioxides and 6 water, is that right? The energy that's released by glucose, some of that energy is captured and then stored and trapped in ATP. And then ATP, when it will be used whenever the cell needs energy. So highly reduced compounds like glucose contain a lot of energy, and we call that potential energy. So let's talk about aerobic respiration, how our cells get their energy needs. I guess our muscle cells also get it from fermentation. We'll talk about that later. Aerobic respiration is the main energy conversion in many cells and is the main energy conversion in all of your cells. In aerobic respiration, we have glucose and oxygen, which is converted by aerobic respiration into carbon dioxide and water. And I have to warn you, aerobic respiration is also called cellular respiration, which is actually a bad term to use because not all cells use aerobic respiration. But the term is cellular respiration means aerobic respiration. I will typically use aerobic respiration when I'm talking about it. Not all of the energy in glucose is converted to the energy in ATP, but some of that energy in glucose is converted to the energy in ATP, and then the cell can use the energy in ATP for its energy needs to perform cellular work. But some of the energy in glucose is lost as heat. And remember, heat is another form of energy. Whenever you convert the energy from one form, like in glucose, converted to the energy in ATP and carbon dioxide and water, they do contain some energy. Whenever the energy is converted, you always lose it. Some energy is lost as heat. And that's just a law of thermodynamics. If you happen to remember from Biology 160, that's actually the second law of thermodynamics. So the summary reaction for aerobic respiration is we start with glucose, C6H12O6. And if you don't know that, you should learn it, plus 6 oxygen molecules, meaning 6 O2s, and 38 ADPs, adenosine diphosphate, plus 38 phosphates, and they're inorganic phosphates, that's why the I, is converted into 6 CO2s 6 carbon dioxide molecules, and 6 water molecules, and 38 ATP molecules. Any question there? This is a summary reaction. Aerobic respiration happens in a number of chemical and physical steps, meaning chemical reactions as well as some physical reactions. A chemical reaction is when a chemical changes from one form to another molecule, like carbon dioxide changing into CO2 and water. A physical reaction is where the molecule changes state, such as when you freeze milk and sugar and cream, and then you get ice cream out of it. That's the physical reaction. Any question about any of that? All right. Some energy from some redox reactions is used by the cell to make ATP. ATP is generated by phosphorylation. Phosphorylation is just the addition of an inorganic phosphate molecule to the molecule. In this case, phosphate and ADP are combined to make ATP. And there we have ADP, adenosine diphosphate with 2 phosphates, plus energy, and then an inorganic phosphate makes ATP. The squiggly line here and there represents a high energy bond. And if the cell wants energy, it can break this bond to release the energy for its energy needs. If the cell wants more energy, it could even break this bond because it has a high energy associated with it also. Any question about any of that? If not, there are 3 main ways that cells can make ATP. There's substrate level phosphorylation, where phosphate from a phosphorylated compound is moved directly to ADP. For example, in glycolysis, we have a 3 carbon molecule. We're not going to talk about it, what it is, but it has phosphate on it, and it can be moved to ADP. And then we have our 3 carbon compound, and then we get ATP. That is substrate level phosphorylation. Any question about that? This happens in glycolysis as well, but we'll talk about fermentation and glycolysis later, probably tomorrow. You know, I don't think I'm going to finish this lesson, but we'll see. The generation of ATP can happen by respiration. Respiration can also be oxidative phosphorylation, but I do not use this term. The respiration involves a sequential transfer of electrons from an electron donor molecule to intermediate molecules to a final electron acceptor. The electrons, in the case of aerobic respiration, are donated from glucose, which then donates it to NADH, or NAD to become NADH. NAD then donates it to FMN, you don't need to know this, and then to Q, then to whatever, down the line. And eventually the last molecule donates the electron to oxygen, to oxygen gas. So we call oxygen gas the final electron acceptor because that's where the electron ends up at. In aerobic respiration, that oxygen is accepting the electrons and the hydrogen ions, and it's converted into water. We'll talk about that later. And then the energy that's released in the different steps is used to generate ATP.
I have a question.
Uh-huh.
Is this the electron transport chain?
This is the electron transport chain, which we will talk about in a few slides. Okay. All right. Let's move on. The third way that cells can generate ATP is by photophosphorylation. This only occurs in photosynthetic organisms, like green plants, these muddy boys over here, green photosynthetic organisms. I'm trying to be a little cute here. Boys are not photosynthetic. What happens is the light releases electrons from chlorophyll, and then the light energy is converted into chemical energy via an electron transport chain. We'll talk about that at the end of the lesson. And then the energy is used to make ATP and NADPH, but these molecules are often used in photosynthesis to make sugar. They don't have to be used, but they're often used up to make sugar. The point is that there are three main ways that cells can make ATP. By substrate level phosphorylation, by respiration, and by photophosphorylation. Any question about any of that? Now note, when I said respiration, I did not say aerobic respiration. We'll talk about the differences between aerobic respiration and anaerobic respiration. I don't know if we'll get to it today or not. We might get to it on our next lesson. We might get to it today. All right, when cells release the energy in molecules, they're released in controlled ways and stored in a series of reactions, meaning the energy released from glucose is not released in a single burst. And for example, if this cup was loaded with glucose and I were to put a blowtorch to it to light the glucose, so much energy would be released at once that I would not be able to. I'm actually sitting right here next to this cup that has burning glucose. And yet glucose is burned in your cells all the time. And the reason why it can happen in your cells is because the energy is released in controlled ways in a series of reactions. And how that happens is the electron is passed from one donor molecule to another compound, another donor molecule, passed to another, and it happens in a series of redox reactions. And then the cell uses that to make ATP. So for our cells, we get our energy needs from carbohydrate catabolism. Many bacteria also get their energy needs from carbohydrate catabolism. And as I stated, even green plants, when they need energy and there is no sunlight available, they will get their energy needs from carbohydrate catabolism. Most organisms use this for their primary source for cellular energy. And the most common energy source for carbohydrate catabolism is glucose. But you may use other molecules. You may digest lipids and you may digest proteins. I don't remember if I mentioned in the class, but a marathon runner actually will start digesting lipids. However, the marathon runner has to be running for 45 minutes before they will turn to digesting lipids for their energy needs. Before that, and that's for 45 minutes, they'll be relying on carbohydrate metabolism or carbohydrate catabolism. Any question about any of that? People who eat a high protein diet and a low carbohydrate diet, they get their energy needs from digesting proteins. To produce energy from glucose, microorganisms use two general processes. And I can actually say that our cells use these same two processes. They can use respiration or they can use fermentation. Now for our cells, all of our cells use aerobic respiration. But our muscle cells can use fermentation to get their energy needs. Okay, and bacteria can use respiration, both aerobic respiration and anaerobic respiration. And bacteria can also use fermentation. Like for example, E. coli can use fermentation. If glucose is available and no oxygen is available, E. coli can use anaerobic respiration. And then if oxygen is available, E. coli can use aerobic respiration. Any questions about any of that? Come on. All right, this will probably take a little while to load, but let me go ahead and get started. And then we'll come back to it. That's an introduction to aerobic respiration, which is what we're going to talk about. Here is showing you a overview of aerobic respiration on the left, and then later we'll talk about fermentation, which is shown on the right. Aerobic respiration has 3 stages: glycolysis, the Krebs cycle, and the electron transport chain, or the ETC. Fermentation only contains glycolysis. There is no Krebs cycle. There is no ETC, or electron transport chain. The ATP production in fermentation is much lower than it is in aerobic respiration, which yields a high yield of ATP. Why wasn't that playing? Oh, I got to click the link.
All cells need energy to stay alive. Usually your cells get their energy through the process called aerobic respiration. The word aerobic refers to oxygen. In aerobic respiration, oxygen interacts chemically with a molecule of glucose. The glucose is broken down into carbon dioxide and water. This process releases energy, which the cell stores in the chemical bonds of ATP. The first stage of aerobic respiration, glycolysis, occurs in the cytosol, the jelly-like part of the cytoplasm. Glycolysis breaks each molecule of glucose into 2 molecules of pyruvate, energizes 2 electron carrier molecules, and produces 2 molecules of ATP. The next 2 stages of aerobic respiration are carried out in the mitochondria. Pyruvate enters a mitochondrion. Once inside, it is modified and enters the Krebs cycle. The reactions of the Krebs cycle break the pyruvate molecules down to carbon dioxide, create 2 more molecules of ATP, and energize more electron carrier molecules. The final stage of aerobic respiration is electron transport. The electron carrier molecules that were energized in the earlier stages deliver electrons and hydrogen ions to the electron transport chain. As the electrons are passed down the chain, energy is released and used to make ATP. This final stage produces as many as 32 molecules of ATP from each molecule of glucose.
All right, any questions?
It started when brain fog nearly got me killed up here at 9,000 feet. So if you're over 40, 50, or 60 years old.
Sorry about that. What am I on here? All right, let's talk a little bit about aerobic respiration. This is another overview of aerobic respiration. We can say that the purple square is a cell, and it's a eukaryote cell because it has a mitochondria. Glycolysis occurs first, where glucose is in a series of steps converted into pyruvate. Glycolysis happens in the cytoplasm. That's why it's drawn here in this cell. And glycolysis does yield ATP. It also makes NADH, and we'll talk a little bit about that later. And it makes pyruvate. So glycose is converted into pyruvate. In a eukaryote cell, the pyruvate then moves from the cytoplasm of the cell to inside the mitochondria. In the mitochondrial matrix, that's inside the inner mitochondria, is where the Krebs cycle happens. And in this slide, the Krebs cycle is called the citric acid cycle. And it yields ATP and CO2. It also yields NADH and FADH2. The NADH and FADH2 from the Krebs cycle and from glycolysis carries the electrons to the electron transport chain, which in this picture is called oxidative phosphorylation. It's also called electron transport chain or chemoosmosis. I will always use the term electron transport chain. I think oxidative phosphorylation is just a little confusing. And in that process, the energy that's carried in the electrons from NADH, FADH2 from glycolysis and the Krebs cycle is converted into ATP. And that conversion happens by oxidative phosphorylation, and we'll talk more about that. Any question about the overview? Now this is showing you aerobic respiration for a eukaryotic cell and where each of the steps occurs in a eukaryotic cell. It's not exactly showing it, but it kind of is. And that is the electron transport chain is happening in and across the inner mitochondrial membrane. And we'll look at that more later. Glycolysis is the first step in actually respiration, and we're talking about aerobic respiration. Glycolysis is also called the Embden-Meyerhof pathway, but I don't use this term. These are the people who discovered it, and it's just too difficult saying their name. And I'm not German, so I could be pronouncing their names wrong anyways. In glycolysis, the oxidation of glucose occurs, and glucose is converted to pyruvic acid. I'm trying to see, there it is there. I knew it was on here someplace. Pyruvic acid is also called pyruvate. They are slightly different molecules. One has hydrogen atom attached to it, the other does not. But we're not chemists, and your cells use pyruvate, the same as pyruvic acid. So we're going to use them interchangeably. And That's because your cell uses them interchangeably. Glycolysis occurs in most living cells. It can occur in the presence or in the absence of oxygen. So glycolysis can happen in a cell that is not engaging in respiration and obviously not engaging in aerobic respiration. Glycolysis does produce ADP and NADH, shown right here, and then the ATP shown there. That NADH takes the electrons through the electron transport chain, and we'll talk about that later. Any question about that? All right, here are the different reactions of glycolysis. When I was an undergraduate, I had to know the names of each of the intermediates from glucose to pyruvate, as well as all of the enzymes in each of the steps. I forget if it's 9 or 10 steps. This molecule is made and then converted to that. And so you can have it going twice, or you can draw it down here, the same reaction. So I think it's fair that because I had to memorize this, that you guys have to memorize that. And I'm sure you all agree with that, right? I guess you do agree. No one's
Please
saying
no. Please no.
No.
I do want you to know that you get a net gain of 2 ATP out of glycolysis. We actually get 4 ATP out of glycolysis, but we have to put in 2 molecules of ATP to get glycolysis to run. So our net gain is 2 ATP. I don't want you to know all of the steps of glycolysis, but you should know that you start from glucose and you end up with 2 pyruvic acids. And then you get a net gain of 2 ATP from glycolysis, 2 pyruvates, also called pyruvic acid, and 2 NADH. Any question about any of that? So that's what you need to know from glycolysis. And you do need to know you're starting from glucose and ending with pyruvic or pyruvate. Let me briefly state that in respiration, there are 2 types: aerobic respiration, which we're talking about now, and anaerobic respiration. In both forms, an oxidation of molecules releases the electrons for an electron transport chain. Usually, the molecule that's oxidized is glucose, and then the electrons are released from glucose and they are sent to an electron transport chain. The difference is in aerobic respiration, the final electron acceptor is oxygen. And in anaerobic respiration, the final electron acceptor is a molecule other than oxygen. We're not going to talk about all the molecules they could be because different organisms use different molecules depending on which anaerobic respiration they are performing. But it could be a nitrogen compound, a sulfur compound, an iron compound. I used to know, is it a carbon compound? Anyway, that's enough. The point is, in respiration, ATP is generated by oxidative phosphorylation. And so the pyruvate or pyruvic acid is goes to the Krebs cycle for the next step of aerobic respiration. However, pyruvate cannot be directly used by the Krebs cycle. So we have the preparatory step, converting the pyruvic or pyruvate or the pyruvic acid into acetyl coenzyme A. So coenzyme A gets added, CO2 comes off, meaning acetyl coenzyme A is a 2-carbon molecule. Pyruvate is a 3-carbon molecule. And then 1 NADH is generated. This acetyl coenzyme A is now ready to enter the Krebs cycle. Please note that the preparatory step runs twice for each molecule of glucose, because in glycolysis, we have 1 molecule of glucose converted into 2 pyruvic acids. Any question about any of that? All right, I should warn you that the Krebs cycle is also called the tricarboxylic acid cycle, because this is one of the first molecules in the Krebs cycle. It's also called the citric acid cycle, or TCA for both tricarboxylic acid or citric acid cycle. And you should probably know these names. I'm not going to test you on it, but in one place you'll hear it with this, in another place you'll hear it as that, and in a third place you'll call it or see it as this. Generally speaking, I call it after the Krebs cycle. It's named after the doctor who discovered it, and your textbook also tends to call it the Krebs cycle. So we both use the Krebs cycle. This is showing you the preparatory step, which we've already talked about. In the Krebs cycle, let me blow this up a little. Acetyl coenzyme A enters the Krebs cycle binding to oxaloacetic acid to make citric acid. You don't need to know this compound. Citric acid is then converted to this compound, converted to that compound, converted to that compound. Whoops, went too far. Converted to this compound, converted to that compound, converted to that compound, and then converted to oxaloacetic acid, meaning we're ending up where we began. And that's why it's called the cycle. The Krebs cycle literally goes in a cycle. And as long as acetyl coenzyme A is coming in from pyruvic acid, from glucose, the Krebs cycle will be running. And hopefully in your cells, that'll happen for as long as you're alive. Because if it ever stops, you're no longer going to be alive. In the Krebs cycle, you generate 1 ATP. That happens down here. You do generate 4 NADH. 1 is in the preparatory step right here. 1 right here, 1 right here, and then the last one right there. You also generate 1 FADH2 molecule right here. And we'll talk about the NADH and the FADH2 a little bit in our next slide. And then we convert all of the carbon in pyruvic acid into carbon dioxide, meaning there's 3 carbons in pyruvic acid. Each of them is converted into a molecule of CO2. Any question about any of that? All right, you should know that both the preparatory step and the Krebs cycle run twice for each molecule of glucose. Because in glycolysis, 1 molecule of glucose is converted into 2 molecules of pyruvic acid. And so if you're talking about how many ATP are generated per molecule of glucose, you would multiply by 2. And the same thing for the number of NADH per glucose. Any question about that? And you can also note that each of the carbons in glucose, there's 6 of them, is converted into 6 CO2. So all of the carbons in CO2 come from the carbon in glucose. Any questions about that? And I guess another point is to say that that means that all the carbon dioxide you breathe out, none of it comes from the oxygen you're breathing in. All of it is coming from glucose. Any question about any of that? All right. Actually, I have it here, so let me go ahead and talk about that. The NADH and the FADH2 carry electrons to the electron transport chain. And we're seeing that here. Let me shrink this a little bit. The electrons from glycolysis and from the Krebs cycle and the preparatory step, which isn't shown here, are carried to the electron transport chain by NADH and FADH2. Any questions about that? All right, so let's talk about the electron transport chain, the third step of aerobic respiration. The electron transport chain is a series of carrier molecules that are each in turn reduced and oxidized as electrons flow from one place to another. They start from glucose, and then NADH2, or excuse me, NADH or FADH2 bring the electrons to the electron transport chain. And then the electrons are moving from one member of the electron transport chain to another. And the electrons are just passed down the chain. Energy is released as the electrons are transferred from the high energy molecules to the low energy molecules. And the energy is used by chemoosmosis to produce ATP. This will probably take a moment to load, so let me go ahead and start. Here we're looking at the electron transport chain, and you can see there are a chain of proteins in a membrane. These right here are actually the electron transport chain. In eukaryotes, this membrane is the inner mitochondrial membrane. So the electron transport chain is in the inner membrane of the mitochondria, the inner mitochondrial membrane. In prokaryotes, the electron transport chain is in the membrane that we call the cell membrane. Any question about any of that? All right, let's watch our little video.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, two mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. 1 hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
All right, any questions on the electron transport chain? If not, let me just summarize it. ATP molecules are produced by the electron transport chain of aerobic respiration. In eukaryotes, they make between 32 and 34 ATP molecules. In prokaryotes, they make 34 ATP molecules. Remember, we're only talking about the electron transport chain. We're not talking about the entire aerobic respiration. The final electron acceptor is the molecule of oxygen. Oxygen combines with the electron and the hydrogen to form water. And that's what happens to all of the oxygen you breathe in. Excuse me. It's converted into water by aerobic respiration. None of it is converted into CO2. It shows you the general summary of electron transport, where the electrons are transported from NADH or FADH2 to a member of the electron transport chain. And then the electron just moves down from one member to another until the electron meets up with the final electron acceptor, which is oxygen. And this shows you the electron transport chain in the membrane. Let me go to the next slide. I mentioned that the electron transport chain is in the inner mitochondrial membrane. That's this membrane of a eukaryotic cell. And so right here, we're seeing the electron transport chain, and that membrane there is the inner mitochondrial membrane. In the prokaryote cells, we still have this electron transport chain, but it's in the cell membrane. Any question about that? Now, for in eukaryotes, when the electron goes through some of the proteins in the electron transport chain, it gives the protein the energy it needs to transport hydrogen from this side of the membrane to that side of the membrane. That happens here, here, and with this protein. For a eukaryote, the hydrogen is transported between the inner mitochondrial membrane and the outer mitochondrial membrane. So the hydrogen is put into that space there. And it's called the intermembrane space. You don't need to know that. In prokaryotes, this membrane is here. And so when these proteins are moving hydrogen from this side of the membrane to that side of the membrane, what they're doing is they're pumping the hydrogen from inside the cell to outside the cell, and the hydrogen is going to be inside the cell wall. That creates a twofold gradient. There's more hydrogen on this side of the membrane than that side of the membrane. The second fold is there's more positive charge on this side of the membrane than on that side of the membrane. So the hydrogen ions, or by the laws of diffusion, going to want to move from this side of the membrane to that side of the membrane. But this lipid bilayer of the membrane does not allow the hydrogen ion to move through the membrane. It can only move through this protein here. And when the hydrogen ion moves through this protein, it gives this protein the energy it needs to make ATP. So this protein that the hydrogen ions move through is an ATP synthase. It converts ADP into ATP. Any question about that? And that's the main way that your cells make ATP. Any question about any of that? As I mentioned, electron transport chain is sometimes called chemiosmosis. I don't use this term. I guess I don't need to talk about that. And the protein gradient here, meaning the higher concentration of proteins here, is called the proton motive force. It's what makes the ATP, meaning there's more hydrogen ion here. They want to move over here. That means there's potential energy right here. And that potential energy is used to make ATP by chemiosmosis. Chemiosmosis specifically is the movement of hydrogen ions, but I don't use the term, so.
Nothing more.
What?
Is it time for the other class?
No, there's 6 more minutes before the other class. I just was trying to come to a good stopping point. And I think I'll stop here. This is the summary for ATP. So I will see you in the lab in 5 minutes now. Are there any questions before I log out? All right, I'll see you later.